Hello and welcome to Nikolai's Genetics Lessons, and today's question is: which of the following concerns about GM genetically modified crops has come true? And you have to choose one of these answers. I'm sure by now you have at least heard the term GMO. Even if you are not quite sure what the term means, you've probably seen labels on the foods that specify non-GMO. There are quite a bit misconceptions and myths surrounding this term and what GMO actually are. But it is important to separate the GMO myths from the GMO facts. Most widespread myth is non-GMO means that the food has not been genetically modified. The truth is virtually all modern day crops have been genetically modified, even the ones that are labeled as non-GMO. While GMO does stand for the genetically modified organism, this term is misleading because it is only referring to one of many genetic modification techniques, transgenesis, and this graph would show you just some of the many different crop modification techniques that have been utilized to create virtually all of the crops available today. Many crops were barely edible or were inedible thousands of years ago. Let's take a look. Traditional breeding. We cross plants and select offspring, and almost all crops undergone this technique. It took thousands of years actually. How many genes would be modified? Anywhere between 10,000 and 300,000. Can you imagine? And we do not consider such plants genetically modified. Next technique would be mutagenesis. We use this technique very widely since we learned about radioactivity. These techniques utilize exposing seeds to chemicals or to radiation, which would cause mutations in such seeds and thus in the whole plant itself and fruits. How many genes would be affected? No one knows, and random changes in genome usually unpredictable. No safety testing requirements. By the way, of course, this first technique also no safety testing requirements. Now let's talk about another technique which we call RNA interference, and in this technique we switch off selected genes with RNA. But actually it would be better to say that we just reduce expression of certain genes with antisense RNA. How it works? We just take messenger RNA and we use reverse transcriptase in order to make complementary strand, which is going to be DNA strand. Then using PCR, we make double-stranded DNA, which we insert into the genome of the plant, for example, and that gene would be able to produce messenger RNA that is going to be complementary to this RNA. And this messenger RNA would be cancelled by another complementary messenger RNA. But we also can regulate to what extent we can cancel this messenger RNA. With this technique, target genes switched off or silenced, or just expression would be reduced, and safety testing is required. Using this technique, only one, rarely two genes would be modified. And next, transgenics. In this technique, we insert selected genes using recombinant DNA methods, and one of four genes can be usually modified. Desired genes inserted only at known locations, and safety testing is required. One more time, take a look. We have many plants that have been modified using these two techniques, hundreds and thousands of plants, and they are not going to be labeled as GMO. Those many thousands of genes would be modified, and we don't know exactly which ones. And these two techniques, only one or two genes would be modified. We know which genes, where they're located, and we do testing, and we have to label such products. An example of artificial selection would be selective breeding of teosinte plant. Take a look. Here on the left, this is how this plant looked just a few thousand years ago, and this is how it looks now. And now we know it as sweet corn. One more time, this is example of selective breeding. An example of mutagenesis would be mutant star ruby grapefruit created through mutagenesis introduced by ionizing radiation. These are both examples of crops that have been genetically modified but are not considered to be GMOs, neither by media outlets or by other modern literature. In fact, there is a database of thousands of mutant crops that have been developed by humans via mutagenesis, and there are thousands of more genetically modified crops that have been created via traditional breeding, as well as many other methods. Yet, as described by US Food and Drug Administration, some popular media and organizations have chosen to narrow the definition, technically incorrect, those becoming more commonly accepted by consumers, to only include a plant, animal, microorganism, or other organism whose genetic makeup has been modified in laboratory using genetic engineering or transgenic technology. But now let's return to our answers, list of answers, or I would say list of myths. First, A. Consumers have experienced allergic reactions and there is no known such cases, so this is false. B. Genes from genetically modified crops have been spread widely to non-GM crops, and actually it is also not true, because when crops are genetically modified, they are modified in such a way that cross-breeding or cross-pollination is not possible. Next statement, there is positive correlation between cancer rates and GM food consumption. And this is also a false statement. There is no such correlation. But answer D, superweeds that are resistant to pesticides have developed, it's actually a concern. It might happen one day, but so far we don't have such weed that is resistant to pesticides. But actually this is a concern, because if we constantly use pesticides, eventually this would lead to natural selection of such weed that is going to be resistant. But so far we don't have such weeds. And genetically modified crops are very effectively resistant to pesticides, unlike naturally occurring weeds. So we have to choose, as you see, answer E, none of the above. All of the above statements are just myths of the different kind. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. And see you in the next video. Goodbye.